we use it to
Kelly hemostat.
We use the tips to grab say the end of a blood vessel where it's bleeding. Hemostat, stop the bleeding. I'm there right now. I'm putting the tip of it into the lateral ventricle. Okay, so the lateral ventricle extends cranially that way, caudally that way. Okay. What I'd like to do is remove this. I'm going to just tear out this corpus callosum. And I want to remove the cerebrum off the diencephalon in here. I don't know the graceful way to do this.
I don't think there's a graceful.
So I want to show you something.
It's like meat.
Okay. So I'm trying to isolate this area here. This is all thalamus here and here, and it projects into the lateral ventricle, which is this space surrounding it. It's kind of a large ovoid structure that makes up the thalamus. Of course, it's bilateral. Now, so if we continue this sagittal section now through the cerebellum, yeah.
Oh, wow.
You look at the pattern
So beautiful.
of the gray and white matter in the cerebellum.
Like
Okay.
nerve fibers, the dendrites.
Looks like a tree.
It looks like a tree, and in fact, this pattern is called the arbor vitae, tree of life, because the white matter looks like, you know, the trunk and branches, and the gray matter, the leaves. Now, we are looking, when I remove the cerebellum, we are looking right down on top of the fourth ventricle, right there. Okay, and remember, it's going to be receiving cerebrospinal fluid through the cerebral aqueduct, which I've got this tip of my hemostat in right here, from the third ventricle, which is, the third ventricle surrounds this thalamus, the lower part of the thalamus. So cerebral aqueduct right into fourth ventricle.
So tiny, everything.
Yeah.
You're really supposed to know what you're doing.
Yeah.
So tiny.
Okay. There is a very thin roof to this fourth ventricle, but because it's so thin, it's easy to cut right through it. Okay, and that's what I've done. Now, let's now take a section this way through the brain. Where am I?
That's right, you skipped it right out of school.
Oh, wow. Okay, so now you can see the pattern of white and gray matter in the cerebrum. So the gray matter is the superficial layers, okay, of the entire cerebrum. That's where all the neurons are, okay? And then we see the white matter then would be their myelinated axons coming away from the neurons into the depth of the cerebrum, where they're going to make many synapses and send impulses all the way down through brainstem into cord. Here again is part of the thalamus, this space. Right, here's a little corpus callosum into lateral ventricle right there. What else can I show you? Why don't I stop there and now follow the directions in lab 32 for the sheep brain. Okay, just follow them carefully, try to identify structures as you go. All right, our presenting group needs to show us 20 structures. So you're going to need more than one brain, probably. And so I would like to ask everybody else, before you throw out the parts of your brain, show them to our presenting group and let them choose what they'd like, okay? Because they're going to need extras for their presentation. Okay, so who's in our presenting group? Okay, these folks. All right. Okay,